my name is Katie Fialos. I'm one of the cancer genetic counselors at Johns Hopkins. So tonight I'll be presenting half of our talk on cancer diagnosis, New Hope Through Genomics, and then so I'll be giving kind of a background on hereditary cancer and genetic testing that will lead into Dr. Armstrong's talk about how that plays into treatment and guiding treatment options for patients with cancer. So hopefully it will be interesting and informative for everyone. So just briefly about me, I graduated back in 2017 from the Hopkins NIH Genetic Counseling Program, and I've been working at Hopkins in cancer genetics since then. Disclosures, they ask us to do all the time. I don't have any financial disclosures. I do have some images in the slides that I give the source for, and several of them are from genetic testing laboratories. I'm not affiliated with any lab or paid by any of them, nor do I endorse anyone specifically, but they do have some good educational materials. So like I said, I'm going to give kind of a brief general overview of hereditary cancer. What is it? What does that mean? Talk a little bit about genetic counseling for hereditary cancer, and then touch very briefly on hereditary breast cancer and hereditary colon cancer. And then talk a little more about panel testing, and at the end I do have a slide of resources if you're interested in more information. So there is a lot that could be talked about in terms of hereditary cancer, so it's going to be very limited in terms of what I can talk about in 20 to 30 minutes, but hopefully it'll be a nice overview laying the foundation. Okay, so starting with hereditary cancer. So the very first important thing to know and to think about is that all cancer is genetic, but not all cancer is hereditary. And in fact, most cancer we know is not due to something inherited. And so cancer we know at its core is a genetic disease. So it's due to accumulations of genetic mutations in the cells. So, and that'll be potentially relevant for some of what Dr. Armstrong might talk about later. So that's an important point to keep in mind. Oh, wrong way, sorry. So just to give kind of a brief explanation, so how can this all be genetic but not hereditary? So here we have an image, and it's probably a little hard to see in the back, but we have, if we imagine here, some cells in our body, and they are showing the nucleus, and then we have two genes that they're highlighting in each of these cells. And then over time, with normal aging and processes, we know that we can have mutations in some of those cells. So then in this one, it shows that in just one of those copies of the gene, a mutation has occurred. And so this is not something inherited, just something that has happened in one of the cells in this individual over time. And then as life goes on, it is possible that that person might develop a mutation in their other copy of that same gene in that same cell. So we all have two copies of every gene. And if both copies are affected in one cell, that is what can cause there to be a lack of regulation, and the cells can kind of start to grow abnormally, and that can develop into a tumor. So this is most of what cancer is. So this is what we would call sporadic cancer, not something inherited. It just happens over time due to a variety of factors. So I have that up top to show, and then the difference with hereditary cancer is, as you can see below, this individual was born with one mutation in one copy of a gene in all of the cells of their body. So all of their cells already look like that one mutated cell that you saw above. So then over time, they could acquire a second mutation in that same gene in the other copy, and then a tumor could go on to develop and cause cancer. And so that's where you can see that there's a much shorter time, and there's less events that we need for a person with an inherited risk for cancer to actually develop cancer versus someone who develops a sporadic cancer. So what we know is that actually we think only about 5 to 10% of all cancers are actually due to something inherited. And often when I talk with patients, they're surprised to hear that. They think that it's more. So up here, the font is pretty small. So we have in that purple wedge, the 5 to 10% of hereditary cancer, and that would be, like I showed, being born with one of those mutations that gives an increased risk. And then the big yellow section is sporadic, and that's what I talked about, where it takes longer to go on to develop that tumor usually. So most cancer, while it's all genetic, most is not hereditary. So when we're thinking about looking at a family or an individual to think, is this someone who may have a hereditary risk for cancer? We look at some things, and the family history is really often key in identifying that. So some of the main indicators for a possible hereditary cancer in the family is someone diagnosed at a young age. So like I just showed in how those cells can go on to change and to form a tumor, since it takes less time if there's an inherited risk, then we often see people diagnosed at younger ages than expected. So if we think about breast cancer, we know that women diagnosed under 50, that's an indication that there might be a hereditary cause, because that's younger than the average age of diagnosis. We also think if there's multiple individuals in the family with the same type of cancer, or cancers that can be related, and then also especially if we see that over multiple generations. And then finally, there's certain rare types of cancer, like male breast cancer, we know is very rare. So when we see that type of cancer, we think maybe this person was born with something that gave them an increased risk to develop that cancer. And then some other indicators we think about are multiple primary cancers in one person. And multiple primary means, for example, one person who's had both breast and ovarian cancer versus a breast cancer that's spread to another part of their body. So a metastatic cancer is different from a new cancer diagnosis. Certain ancestries we know have increased risk of having a hereditary cancer. And then also sometimes certain characteristics of the tumor could indicate that there might be a hereditary cause as well. So in terms of how these hereditary cancer syndromes are inherited, so first thing to know is that we all have these cancer genes. So for example, BRCA1 and 2 are some of the ones that people are most familiar with. We all have them. We all have 2 copies of them. The question is there an alteration or a mutation in one of those genes that causes an increased risk? Okay, and I put up here, so I'm using the word mutation, and that's generally the word that most people have heard. The genetics community is trying to move away from that so much. So I've included in here some other terms. So pathogenic variant is now the technical term, but that can be a mouthful. So I often talk about genetic alterations when I'm talking with patients. And then these are passed down in a way that we call autosomal dominant. And what that means is, like I said, we all have 2 copies of the gene. And when something is dominant, it means that having an alteration in just one of those copies is enough to give the increased risk. So like I showed before, when people are born with that alteration in one copy, we saw that then it takes a shorter time for them to develop cancer. So that's the increased risk that they're born with. And then also because of that, we only pass on 1 copy of each gene to our children because they get the other copy from their other parent. Someone who has a mutation in one of these genes, each of their children would have a 50% chance to inherit that. And then there are a couple myths that people think about how these hereditary cancer syndromes are inherited. So I've heard from patients, and some have even said that their doctors have said to them that only the maternal side matters in terms of thinking about, for example, a woman's risk for breast cancer. And that's not true. So men can carry these genetic alterations as well, and because they have much less breast tissue, they don't have ovaries, they may never develop cancer themselves, but can pass on that hereditary risk to their daughters. So that comes back to thinking about both sides of the family history are really important when thinking about whether someone has a risk for hereditary cancer. So this is just an example of a family history, or what we would call a pedigree in genetics. And so we use this routinely when we're talking with patients and thinking about whether they have risk for hereditary cancer. And here you have, so on these drawings, the women are circles and the squares are men. And so you'll see that we have several of those characteristics that I mentioned that can indicate a hereditary cause for cancer. So we see 3 women diagnosed with breast cancer in multiple generations, and then we have 1 diagnosed very young at age 34, and we also have prostate cancer and ovarian cancer in the family. And we know that all of these types of cancers can be associated with alterations in the BRCA1 or 2 genes. And then here, like I just talked about, you can see for this young woman who has breast cancer at 34, it looks like if there is something, she inherited it from her father. So it's really on her paternal side that we see this increased family history of cancer. So it's important to look at both sides of the family. So that's kind of my brief introduction to cancer genetics and hereditary cancer. So then in genetic counseling for hereditary cancer, what would we do? And so some things we do is similar to like I just talked about with patients. I review their personal and family history, and that helps me to get a picture of what do I think the chances are there would be a hereditary cancer syndrome or a risk for cancer in this family. And then based on that, I would discuss options for genetic testing if it's appropriate. So based on that assessment, does this person need to have genetic testing? And then if the individual wants to have testing and we think that it does make sense for them, we would go through the consent and we could get the sample to do testing, which can be either blood or saliva. And then another important piece to genetic counseling is addressing the patient's concerns about genetic testing and providing support. And this can be a variety of different things. So, some patients might be very anxious about the types of results that they could get back, what they might learn from this testing, what the implications would be for family members. For a lot of patients, it's worrying that they might have passed something on to their children who would then be at risk and what that would mean. And then some people have concerns about what a result could mean for their insurance or their ability to get insurance in the future. So this is, we have a pretty broad conversation, not only about the science, but also about how this could impact the patient. And then it's important to know that you can see a genetic counselor without having genetic testing. So sometimes patients want to come in and they just want to get that information and know, what is the science? Are they at increased risk? Do we recommend testing? And then they want to make a decision about whether or not they want to have testing. So certainly we welcome patients that are interested in just talking with us for a consultation without necessarily being committed to testing. So very briefly, I'm going to touch base on the insurance concerns that I mentioned, because this is something that I hear a lot of from patients, and I've heard from some people that, they don't even want to go see a genetic counselor because they're worried about this. They think they just don't want to have testing. So it's important to know that in 2008, there was a federal law passed called the Genetic Information Non-Discrimination Act, better known as GINA, because that's easier to say. And this is completely separate from the Affordable Care Act. So regardless of whatever may happen to that law in the future, this is a separate law. And it essentially says that employers with more than 15 employees and health insurers cannot use a positive genetic test result alone to discriminate against the individual. So that means you can't be fired from a job and you can't be denied health insurance based on a positive genetic test result. It does not apply to members of the military and some federal employees, and those groups do have their own protections. So the one part that we talk to patients a lot about, so it's good that there are those protections, and for a lot of people that is reassuring. However, the law doesn't actually say anything about life insurance, long-term care insurance, or disability insurance. So what that means is that if someone does test positive and then down the road goes to take out a new life insurance policy or expand an existing policy and they have to qualify, as part of all those medical questions that they ask, they can legally ask about genetic test results, and they can take that into account. Whether they all are or how they're using that information is more of a question. There's just nothing in place that says that they can't. And so often we'll talk with patients about this, especially younger patients who maybe haven't had cancer, and they'll think about if they want to put any policies in place or change any policies before they have testing. Okay, so then very briefly, hereditary breast cancer, and there are many genes that we know of now that can be associated with increased risk of breast cancer. I'm mostly just going to focus on the main ones due to time, but I'll give you kind of a flavor for what else is out there. So BRCA1 and 2, sometimes also referred to as BRCA1 and 2, these are two genes, sometimes called the breast cancer genes. They're two completely independent genes. So they are called BRCA1 and BRCA2 because they were first identified for their association with breast cancer. That's what the BRCA is for. But in our bodies, even though they have similar risks, they work independently. So someone can have an alteration in their BRCA1 gene and their BRCA2 gene is entirely normal. In fact, that's usually what it would be. It's very rare to have alterations in both of them. They were the first hereditary breast cancer genes identified in the early to mid 1990s, and they still account for the majority of hereditary breast cancer that we identify, about 50 to 75%. And that's of hereditary, so only of that 5 to 10% that we find a hereditary cause, this accounts for about half of that. Wrong button. There we go. Okay. So we think that about 1 in 300 to 1 in 400 individuals in the general population actually has a mutation or an alteration in one of these two genes. Yes, sorry, that got separated. But it is much higher in certain ethnicities. So we know that individuals of Ashkenazi Jewish ancestry, we think it's actually about 1 in 40 individuals who has one of 3 specific founder mutations. And then they both do give increased risks of breast, ovarian cancer, as well as pancreatic and prostate cancers. And then BRCA2 also has an increased risk of melanoma. And there can be increased risks of male breast cancer as well, especially with BRCA2. So to give you kind of a picture of that, this is a resource from Myriad, which is a genetic testing company. And so you can see here they group together in purple. We have the risks associated with a BRCA mutation. And here, even though they're separate genes, they're grouping together BRCA1 and 2. And then in the blue, we have what's the risk for the general population. And at the bottom, if you can't see it, we have breast cancer by age 50, then breast cancer by age 70, followed by a second breast cancer, and then ovarian cancer. So you can see that there's significantly increased risks with these two genes. And I will say, here they're saying up to, so these are the very highest percentages that have ever been found in any study, and a lot of studies have put that lower. So, for breast cancer, it's maybe more closer to 50 to 70% depending on the gene. But some studies certainly have shown risk that high. And then here, this just gives you a little bit of an idea of the difference between BRCA1 and 2. So we have BRCA in the teal and then BRCA2 in the red. So here again, for the breast cancer, it shows them being pretty similar. Again, with kind of the maximum lifetime ever. BRCA1 does have a bit of a higher risk of breast cancer and then definitely a higher risk of ovarian cancer. You'll see that there. But then, notably, BRCA2 has higher risks for things like pancreatic, prostate cancer, and male breast cancer. So for BRCA1, we don't even necessarily always have good numbers for that. And so, some people will ask me, well, which one's worse? And it kind of just depends how you look at it. Certainly the risks are different. And then this, like I said, I'm just going to touch on briefly, gives you kind of an idea of the other genes that have since been identified to be associated with increased risk of breast cancer if there's a mutation in one of them. So you'll see that we have some genes that have significant risk, almost as high as BRCA1 and 2, and then we have others that are much lower. And then you'll also notice that some of them, so they're here, the part that's shaded, they're giving you the range. So for some of these genes that we've identified more recently, we have a very broad range. So it's not clear yet exactly how high that risk really is. All right, so then I'm going to move on to hereditary colon cancer. And again, I'm going to basically mostly talk just about the main player there, which is something called Lynch syndrome. And so this is due to alterations or mutations in one of 5 specific genes. So in this case, there's actually 5 different genes that can cause Lynch syndrome. And the highest risk is for colon cancer, followed by uterine, or also called endometrial cancer in women. And then there are basically a smattering of increased risks, but lower than these two, colon and uterine. And those are things like ovarian, small bowel, stomach, urinary tract, pancreas, etc. And so we think all of those risks are about 10% or less. And then pretty similar to BRCA1 and 2, we think about 1 in 300 individuals are estimated to have Lynch syndrome. And we think that in this case, it's responsible for about 3 to 5% of all colon cancers that are diagnosed. So this one, I know with the screen, it's kind of hard to see. But here, the main point is they're showing the different cancer risks, and they've really broken it out by gene. So I mentioned that there's 5 different genes that we know that an alteration in one of those causes Lynch syndrome. But essentially, kind of the type of Lynch syndrome you have can be very different depending on the gene. So they've grouped together 2 of them, MLH1 and MSH2, and actually also EpCAM, and those account for the majority. About 80% of people with Lynch syndrome will have a mutation in one of those genes. And the risks are certainly the highest in those for colon cancer, endometrial, and then for the other cancers, they really only indicate the risk for those. But then you'll notice, for example, in red, we have PMS2, which has been identified much more recently, and while the risks are elevated over the general population, they're much lower than the others. So it's all called Lynch syndrome, but the actual risk that an individual has can be very different depending on the gene. So then I'm going to talk very briefly about panel testing, and this has certainly become the norm in the last 4 to 5 years, I would say. So this is just an example from a laboratory called Invitae, and this gives a kind of a picture of all the different hereditary cancer genes that you can be tested for. So they have it broken down into different panels, and so their smallest here that they show is 7 genes, and then you can get, if you get all of the genes tested, you can actually get up to 84 genes tested that are associated with different forms of hereditary cancer. Not necessarily all breast and colon. We have genes on here that are associated with ovarian cancer, pancreatic cancer, skin cancers, a lot of different things, renal cancers. So this is really expanding and it's certainly become much more common. Used to be the norm to test for BRCA1 and 2, and now we're definitely testing for more and more genes. So testing for between 30 and 40 genes is not uncommon. And that brings me to the point of when I see patients who say, oh yeah, I was tested before and I was negative, or my mom was negative. And so here, for example, these, you can't see them, unfortunately, very clearly. These are all negative BRCA1 and 2 tests, and they are all testing for very different things. So we have someone who it seems was tested for one specific mutation that was probably in the family. Someone was tested only for those common in the Jewish population. So there can really be a big difference even between just what you were tested for BRCA1 and 2. Oh, sorry, wrong button again. Yeah. Not to touch. Oh no. I think I made it worse. Okay, here we go. So, and then here you'll see, so an example down at the bottom, someone who was tested for about 9 genes associated with breast cancer versus this panel probably has about 30 on it, and that one up there has, you know, probably the 80, 83, 84 genes, something like that. And so when patients tell me that they tested negative or their relative tested negative, my question is negative for what? And so it's really important if you or someone in your family has had genetic testing to have a copy of that test, because even if it's negative, knowing what was tested is really important to know what has been ruled out and what might still be out there. Oh gosh, sorry. The buttons are so close together. Okay. And so that leads to updated testing. So now we're seeing many patients back who had previous negative testing, and we're seeing them back because before, like I said, it was common to only test for 1 or 2 genes at a time, and now we're regularly testing for 30. And so especially for individuals who, have an indication for genetic testing, if they had negative testing before, say, 2015 or so, they should probably consider additional testing. And if someone has questions about whether they or someone in their family should have more testing, I always say ask a genetic counselor. And that's where it's especially helpful to have that report to know exactly what was tested before. So then here at the end, I just have a list of kind of some resources. Again, it might be hard to see in the back, but I have at the top the contact information for our clinical cancer genetics and prevention service, and then some more information on that GINA law if you have any questions. The National Society of Genetic Counselors has a good patient page giving more information about genetic counseling and related topics. And then FORCE is a great organization that works with individuals with hereditary breast cancer. And Alive and Kicking is a newer organization, but they seem to be doing some really good work in terms of Lynch syndrome advocacy. So if you have any nagging questions, those are some good places to go. And then I'm going to turn it over to Dr. Armstrong.
So I'm going to talk to you about something that's, I think, probably the easiest way to discuss it is as a parallel universe, which is genomics. And while genetics and genomics certainly talk to each other and have a lot in common, they actually are quite different. And we'll go through some of that. So I'm going to talk about genes in cancer, identification of molecular and genomic alterations for cancer treatment, how genomic testing is done, and then if we have time, we'll talk specifically about some of the implications of genomic testing for, kind of 2 new treatments, new therapeutic approaches that we have over the last 5 to 10 years. One is immunotherapy, and the other is PARP inhibitors, and then genomic initiatives and some clinical trial implications. So, Katie talked a little bit about this on the left. The mutation, which is indicated in yellow, is present in all cells in the body, and that's an inherited cancer. But you can see here the little starburst with yellow is the cancer that formed from that. On the right, in somebody with a somatic mutation, they can actually, in the cancer, have that same mutation, but it was not inherited. So it's not there in all cells of the body. So it occurs in non-germline tissues. It won't get passed on to children. It's not heritable. And some of the first things that we learned about tumor genomics were things that we then used to test for hereditary cancers. We now are learning things about hereditary cancers that are leading us to test tumors, even in patients who didn't inherit those. So these worlds sort of talk to each other, but they don't talk to each other very well at this point in time. So we'll talk a little bit about that. So as we talked about, the same gene can be mutated in a sporadic cancer or in somebody who has a hereditary cancer. And the P53 gene in the Li-Fraumeni syndrome and the APC gene in familial adenomatous polyposis coli syndrome were 2 of the first that were identified initially in tumor and then were found to be germline mutations in patients. We now are doing a lot more tumor genomic testing than we are germline testing, and we fully expect that tumor genomic testing will identify genes of interest for germline testing. And I think so we'll probably move a little bit more toward that. And we can talk about that later if we have time. So genomics is the branch of molecular biology that's concerned with the structure, function, evolution, and mapping of genomes. It involves the study of specific genes, parts of genes that have a known function. And the genomics is the study of the entirety of the organ's genes to find variations that affect health, disease, or drug response. And it is a newer field than genetics. Cancer genomics focuses on the difference between tumor cells and normal cells. And many of the studies that we've done have specifically looked at that to identify these genomic alterations. This is just kind of taking us back a little bit to molecular biology 101. What you can see on the top is a gene. The gene has many parts to it. The blue parts, which we call the exon in the DNA in the top line, will get transcribed and turned into RNA. And then that RNA will have those little red parts spliced out, and you will end up with a second type of RNA shown just in blue, which then gets changed into a protein. It gets translated into a protein. And just to keep this in mind, that down here at the bottom, these proteins, we have been testing for decades in tumors. We have been looking for changes in these to drive and to lead us in terms of how we treat our cancers. So many parts of this we've been doing for a long time. What's new is looking at RNA and DNA specifically in the tumor and how that's driving our therapeutics. So this is just a few things that we have been doing for a long time. The estrogen receptor, the progesterone receptor, HER2/neu. I treat breast cancer, so I'm focusing on those, but we've been testing those for decades, and they have driven our therapy. And they really are a type of genomic testing. But they're a little more old school. We still do them. They're very reliable. They're very well established. But now we can look in different ways at them and others. And I included a couple on the bottom. Katie talked about the DNA mismatch repair proteins in Lynch syndrome, which are now driving therapies, PD-L1, and some others. So this is actually a limited list of cancer biomarkers. I interestingly just picked these up from a paper, and as I was going through it, I said, this paper has to be 2 or 3 years old because some of the things I know that we're using today were not in this paper. But interestingly, when I studied for boards about 3 years ago, this list in lung cancer, which you can see is pretty long, was pretty much what I studied and is completely out of date today. So things have changed very rapidly. So what you can see, and I've just chosen a few diseases here, there are, and then in GI cancers there's some subgroups, lung cancer there's some subgroups, but there are these markers, and there are certain tests we can do for those markers that basically dictate those treatments in the very right-hand column. Now, in that test, you'll see a couple. One's called IHC, that's immunohistochemistry, and that's the pathologist in the pathology lab looking for the expression of proteins. Okay, that's a very typical thing that's done. Estrogen receptor is done that way, progesterone receptor, and others. There's also something called FISH, which is fluorescence in situ hybridization. That's been done for a couple of decades for many of these. But what you'll see is there are some of these newer tests that require something called sequencing. And that's where we get back to that DNA. We have to know what's going on in the DNA to know whether these things have been altered and to drive the therapy. Okay, so even when I was studying for boards a few years ago, I had to know about some of the sequencing. Many of these tests can be done through multiple processes, but this is now sort of the map for molecular targets in lung cancer. And I'm going to tell you that 20 years ago, the only one of these things that drove therapy was that very top left one, EGFR, the epidermal growth factor receptor. And now today we have 15 or 20 things that can drive treatments in lung cancer. And as I said, even when I was studying for boards a few years ago, many of these things were not on the list of things I had to know. This is just from a thing called Up to Date, and I put it in here because it talks a little bit about the methods that we use. And I use non-small cell lung cancer because it probably is the one where we have the most things we want to look at of any disease. So for screening non-small cell lung cancer patients for driver mutations, the methods are continually evolving, and there's no single standard platform for testing. The things that would make a test something a provider, a doctor want to order would be, it has a fast turnaround time. You don't want to wait 3 months for that result before you decide what treatment to do. So we like 2 weeks or less. You want it to be cost efficient. You don't want it to cost $40,000. You want it to be covered by insurance. And you want it to be able to be performed on clinically available specimens. If you need a ton of tumor and somebody only has one small site, you're not going to be able to do this. This sequencing, which we now call next generation sequencing, or NGS, can be performed in lieu of some of those standard pathology tests like IHC and FISH, or it can be done in parallel. So you say, okay, I have some stuff that I can't do with IHC or FISH, and so I'm going to send off for that while I'm doing the IHC and FISH. Or I can say, I'm going to wait and see what the IHC and FISH show, and then if I need more information, I can do the test. There's lots of different ways to approach this, and a lot of it will be dependent on how fast the turnaround is in your own pathology lab, and et cetera. So there's different approaches. One of the newest things is something we call a liquid biopsy. And this is basically sort of moving from the usual approach where you have to have a piece of the tumor to send off for this testing. And that's been what's been traditional. Either a tumor was biopsied or was removed surgically. And you take that from the pathology lab and you send it out for this testing. We can now do these blood-based tests that are called liquid biopsies. And they're based on the fact that either cells from the tumor or the DNA from the tumor circulates in the bloodstream. And so you can do this. You can obtain tumor cells or tumor DNA clearly in a less invasive manner. You don't have to put a needle into the organ. We also know some sites of tumors are very risky to biopsy. You know, if somebody has a recurrent lung cancer that's right next to the heart, that's going to be tough to biopsy. It's also usually less expensive than a biopsy, and frequently is done more quickly. It also will reflect the current status of the tumor. So if somebody had a lung cancer 2 years ago and you knew the molecular profile of it, but now they have a recurrence, that tumor can change with time. And so one of the nice things about doing this is you can pretty easily, pretty quickly, not too expensively and not too invasively get knowledge of what does this cancer look like now. And so that's one of the clear benefits. We've done this a lot in lung cancer, and we're moving toward doing this in other cancers. We're doing some of this in breast cancer and in ovarian cancer as well, and in other cancers. So I just want to talk a little bit about how this explosion in molecular and genomic profiling has really driven one of our newest and most successful therapeutic approaches over the last 5 to 10 years, which is immunotherapy. And I'm going to step back a little bit and just talk a little bit about the immune system in cancer. So we know that there's sort of one school of thought, which is cancer is a failure of the immune system to recognize these cells as foreign. Okay, these cells are not normal cells, but our immune system is not recognizing them as foreign. The other side is, yeah, but they came from our normal cells, and we don't want to recognize our normal cells as foreign. When that happens, we get diseases like lupus, rheumatoid arthritis, ulcerative colitis, and those can be quite devastating. We also know that we have an activating and a suppressive arm of the immune system, and we need both of those. But we now can define certain cells of the immune system, lymphocytes, that are in that suppressive part and certain parts that are in the active part. And we can look at them and we can measure them and we can know how many of the activating immune cells and how many of the suppressive immune cells are present. And many studies will show that if you look at the immune cells, the lymphocytes that are in a tumor, they're almost all suppressive. They're actually shutting down the immune response that we might normally have to the cancer, okay? Those are things called tumor-infiltrating lymphocytes, or TILs, and they frequently are the suppressive part of the immune system. So we know that the immune system can control and eradicate cancer. In the setting of malignancy, there's multiple mechanisms that for the our immune system to prevent development of active anti-tumor immunity. So it's those infiltrating cells, it's cytokines that get released and things like that. But we now have identified those suppressive cells, and we have made antibodies to those suppressive cells. So we can suppress the suppressive part of the immune system and start to shift that balance to an active immune system. And this is just a little cartoon showing some of the things. The cancer cell or the tumor cell is in the upper right, and then we have the other 3 cells which are part of the immune system. And these things in red are suppressive parts of the immune system. And they're things called PD-1, PD-L1, which is programmed death ligand 1, CTLA4. Those are the 3 that are the ones that we have most of the drugs that target. And we have these antibodies that are listed here, and they basically block those and they prevent those interactions, okay? And so we can these are many of the different ways we can suppress the suppressive part of the immune system. I will ask, how many of you have seen commercials for Opdivo on TV? Yeah, almost everybody has, and that's one of the drugs. It's one of the first ones that was approved. It's a name drug called nivolumab. So Katie talked to you a little bit about Lynch syndrome and these DNA mismatch repair proteins. Well, it turns out that these DNA mismatch repair proteins, when they're not working, allow the cancers to make proteins that are not considered normal. Our body never saw them before. Our body doesn't think of them as normal. And so in tumors that have what we call microsatellite insufficiency, that they actually will make these new proteins, and they will basically be much more immunogenic. Okay? There's lots of ways we can test for this, but most microsatellite instability, it turns out, doesn't come from Lynch syndrome. It is sporadic in the tumor. It's not inherited. It's just part of the cancer-causing formation. Now, one of the first studies that looked at just the presence of microsatellite instability as a potential predictive response to immunotherapy was done by Young Lee at our institution. And she said, okay, well, we know that colon cancer has Lynch syndrome. And so it has this microsatellite instability. It makes these abnormal proteins, and it may respond. And so let's do this MSI, or mismatch repair deficient colorectal cancer. That's the first column. We're going to test that. And then as a control, we're going to test colon cancer that doesn't have it. And then as sort of an afterthought, she said, well, there's other cancers that have this mismatch repair. Let's just sort of take any of those and we'll test them. And probably not surprisingly, when you look at that objective response rate, it was 62% in the patients who had MMR deficient colon cancer, 0% in colon cancer that was MMR proficient. But in the MMR deficient non-colorectal cancer, this is the first time it was shown that if you had this pattern in something besides a colon cancer, exactly the same response rate, 60%. Okay? And so that really led to kind of a free-for-all of testing in this genomic testing to look for these abnormalities. And this is a study that Young Lee led at our institution, looking at one of these tumor genomic profiling companies and looking at over 12,000 specimens and saying, what percentage of these have microsatellite instability? And you'll see here over the right, it's like 1 or 2% in most of these cancers. But on the very left, shocking to most of us, is a tumor I treat, which is endometrial cancer. And in this study, it was 18% because they took all comers. But in the typical endometrial cancer, it's actually around a third of them will have microsatellite instability. The next ones are largely GI cancers, colon, gastric, and then some others. But you can do these tests for microsatellite instability, and these can now be used to drive immunotherapy. There's other things besides this microsatellite instability. We know that tumors that have a lot more mutation, usually because they've been exposed to carcinogens, and the two biggest ones are lung cancer, because carcinogens like tobacco smoke cause lung cancer, and melanoma, because the carcinogen UV light causes those cancers. They have some of the highest tumor mutation burdens, and they are some of the ones that have the highest response rate to these. We can look at some of these like that PD-L1 expression. We can also look at cancers that have a foreign antigen, and the biggest one is the human papillomavirus associated with cervical cancer, other genital cancers, and now expandingly in head and neck cancers. And those that have a foreign antigen, the body can recognize them as foreign if we help the body to do that with these medications. This is just, I'm not going to go through this. This is looking at this tumor mutation burden, and you can see that some of these cancers they're listed here, but some of them have what we call a high tail up toward the top. That means a high percentage of them have really high tumor mutation burden. And not surprisingly, lung cancer, melanoma, and my favorite endometrial cancer here have these high tails. So I want to talk a little bit more about a completely different approach, which is PARP inhibitor therapy. PARP inhibitor stands for poly-ADP ribose polymerase, and that's why we call it PARP. It is a drug that blocks the repair of DNA damage. That doesn't sound good. We need to repair our DNA damage, right? Well, some cells have a mutation in something called the homologous recombination pathway, and that includes the patients with a BRCA mutation. And these are two parallel pathways for repairing DNA damage. And the cancer cells can't repair that with that HRD or the BRCA pathway. And when we inhibit the ability of them to repair through the other pathway, the PARP pathway, cancer cells die. The normal cells still have that other pathway to back up. And so this has been something we call selective lethality, where you can use this treatment approach and it's got much more toxicity to the cancer cells than to the normal cells. This was an early study that looked at BRCA-associated cancers and showed that these PARP inhibitors truly worked well in BRCA-associated cancers. This was a study that was just reported last year, and I don't want to get into the weeds too much with it, but it was patients with ovarian cancer who had a BRCA mutation. And after they were done with initial therapy, they were randomized to get a PARP inhibitor or not. And this is their recurrence curves. Apologize. And what you can see here is at 3 years, the people who got the PARP inhibitor, less than 50% of them had recurred, whereas only 30% of the patients who didn't get the PARP inhibitor had recurred. The recurrence risk was cut by more than half. And, are some of these patients now going to be cured? These are all people with advanced disease. We don't know the answer because we don't have long-term follow-up. But this led to approval in this situation for these patients. Now, interestingly, we talked about the fact here in green that, as Katie said, about 15% of ovarian cancer patients will have a BRCA mutation. About another 10%, taking us up to 25, will have a somatic or a genomic mutation or alteration in BRCA1 or 2. So that gets us up to about 25%. There's another 25% of patients, so in the blue or purple, that gets us up to about 50% that have something wrong with that pathway. It's not a mutation in BRCA1 or 2 in the tumor or the germline. And the company that makes this HRD assay says, it now predicts this, but it's not all that good. We need better assays, and genomics will take us there. It will show us which of these patients potentially have this, even if they don't have the BRCA mutation. And so this is a very active area of research in ovarian cancer at this time. So I just wanted to stop to talk a little, finish up talking a little bit about some of the really big initiatives. Probably the biggest one over the last decade or so has been the Cancer Genome Atlas, TCGA. And they have now studied 33 different separate cancers, looking at paired tumor and normal in over 20,000 patients with cancer. It's been a yeoman effort. It's led to incredible discoveries. They now are really rich databases for researchers, and we can use these to help us define treatments. I will tell you that this is your tax dollars at work. The Cancer Genome Atlas is through the National Cancer Institute, the National Human Genome Research Institute, the Cancer Characterization Initiative, and then there's studies looking at these associated with HIV-associated cancers. Anyway, there's lots of different national organizations that are specifically looking at how these things can help us drive treatments. There's also a thing called the exceptional responders, and I've got a couple patients in this. They're patients who, in a disease where you expect people to live 4 years are living 10 or 15 years. And now they've asked those patients to volunteer to let their tumor be examined to see what it is about their cancer or what it is about them that's made them respond. And so there's lots of these exceptional responder studies that are going on. These things can lead to treatments that might not otherwise be available for a specific disease. And this is a thing called the MATCH study, which stands for Mutations and Therapeutic Cancer Treatment. I forget exactly what the MATCH stands for. But basically it says if you have a mutation and there's a drug that targets that mutation, we don't care what cancer you have. If you have that mutation, we're going to allow you to be in this study, and we're going to test your tumor, test you for whether you respond to that tumor. And this has been hugely successful. And so, you might have something like a HER2 mutation that's really common in breast cancer, but is 1 or 2% in some other cancers. Does HER2 targeting work in those cancers? You can find out through these kinds of studies. You can also have certain types of mutations that are common in certain diseases but are only a small fraction of other diseases, and you can determine whether those other diseases respond well. And I've got a couple patients on this study. We can do this in many ways. The first one is where you sort of say, I've got this treatment and I'm going to study it in disease A, B, C, D, and E. The second one is I'm going to say this is a treatment where these certain mutations drive it, and I'm going to look at those mutations in certain cancers. And then the last one is basically where I'm saying, I've got a certain drug. If you've got the mutation, you can be in part of that. And so these are called basket or bucket trials, and there's lots of them going on right now. And I will say that, so the fruition of some of this is over the last 2 years, we have had 3 different approvals through the FDA of drugs that are what we call tissue agnostic, meaning they're not approved for a certain disease, they're approved for a certain genomic alteration. So the first one is pembrolizumab, which is one of those immune checkpoint inhibitors, is approved for adult or pediatric patients who have that microsatellite instability. Doesn't matter what kind of cancer you have, if you have microsatellite instability, pembrolizumab is FDA approved for you. The second, last year, was a drug called larectinib, which is approved for a certain mutation called an NTRK mutation. There's 3 different, they're actually rearrangements or fusions. And then this year there was another drug that targets that same fusion. So we are sort of on the cusp of this, you know, we're going to treat the mutation in your cancer, we're not going to treat the name of your cancer. And it's what drives your cancer, and so it will drive your treatment. And that's really kind of, we're really poised on the edge of that right now because of these studies that have gone on. So I'm going to end there, and Katie and I will be happy to take your questions.
Yeah, so the question was for a patient who has had negative genetic testing, their siblings being able to be tested versus nieces and nephews. So I guess, I don't know if I just need to hold that. So, you know, I think if the question is based on your diagnosis, if you've tested negative, we don't necessarily think that there's an indication for anyone else to be tested, because we haven't identified anything hereditary in the family. And so I think as to whether or not one or the other could be tested based on insurance isn't so much the question as who needs to be tested. And I think if you've had, you know, kind of all the genes that could be associated with your cancer and they're negative, then either it falls into that sporadic cancer or there's something else we haven't yet identified. But regardless, we wouldn't be able to find it in one of your relatives.
We always, if we can, when we see, because Katie and I work together, if there's somebody in the family with cancer, we like to test that person first, because if they don't have the mutation, it didn't cause their cancer, then that probably means other people in the family aren't at risk. Thank you very much.
Thank you.